Protein structure prediction. Protein structure prediction is the inference of the three-dimensional structure of a protein from its amino acid sequence, that is, the prediction of its folding and its secondary and tertiary structure from its primary structure. Structure prediction is fundamentally different from the inverse problem of protein design. Protein structure prediction is one of the most important goals pursued by bioinformatics and theoretical chemistry. It is highly important in medicine, for example, in drug design, and biotechnology, for example, in the design of novel enzymes. Every 2 years, the performance of current methods is assessed in the CASP experiment, Critical Assessment of Techniques for Protein Structure Prediction. A continuous evaluation of protein structure prediction web servers is performed by the community project CAMEO 3D. Protein structure and terminology. Proteins are chains of amino acids joined together by peptide bonds. Many conformations of this chain are possible due to the rotation of the chain about each C-alpha atom. It is these conformational changes that are responsible for differences in the three-dimensional structure of proteins. Each amino acid in the chain is polar, i.e. it has separated positive and negative charged regions with a free carbonyl group, which can act as hydrogen bond acceptor, and an NH group, which can act as hydrogen bond donor. These groups can therefore interact in the protein structure. The 20 amino acids can be classified according to the chemistry of the side chain, which also plays an important structural role. Glycine takes on a special position, as it has the smallest side chain, only one hydrogen atom, and therefore can increase the local flexibility in the protein structure. Cysteine, on the other hand, can react with another cysteine residue and thereby form a cross-link stabilizing the whole structure. The protein structure can be considered as a sequence of secondary structure elements, such as alpha helices and beta sheets, which together constitute the overall three-dimensional configuration of the protein chain. In these secondary structures, regular patterns of H bonds are formed between neighboring amino acids, and the amino acids have similar phi and psi angles. The formation of these structures neutralizes the polar groups on each amino acid. The secondary structures are tightly packed in the protein core in a hydrophobic environment. Each amino acid side group has a limited volume to occupy and a limited number of possible interactions with other nearby side chains, a situation that must be taken into account in molecular modeling and alignments. Alpha helix. The alpha helix is the most abundant type of secondary structure in proteins. The alpha helix has 3.6 amino acids per turn with an H bond formed between every fourth residue. The average length is 10 amino acids, 3 turns, or 10A ring, but varies from 5 to 40, 1.5 to 11 turns. The alignment of the H bonds creates a dipole moment for the helix with a resulting partial positive charge at the amino end of the helix. Because this region has free NH2 groups, it will interact with negatively charged groups such as phosphates. The most common location of alpha helices is at the surface of protein cores, where they provide an interface with the aqueous environment. The inner-facing side of the helix tends to have hydrophobic amino acids and the outer-facing side hydrophilic amino acids. Thus, every third of 4 amino acids along the chain will tend to be hydrophobic, a pattern that can be quite readily detected. In the leucine zipper motif, a repeating pattern of leucines on the facing sides of 2 adjacent helices is highly predictive of the motif. A helical wheel plot can be used to show this repeated pattern. Other alpha helices buried in the protein core or in cellular membranes have a higher and more regular distribution of hydrophobic amino acids, and are highly predictive of such structures. Helices exposed on a surface have a lower proportion of hydrophobic amino acids. Amino acid content can be predictive of an alpha helical region. Regions richer in alanine, A, glutamic acid, E, leucine, L, and methionine, M, and poorer in proline, P, glycine, G, tyrosine, Y, and serine, S, tend to form an alpha helix. Proline destabilizes or breaks an alpha helix but can be present in longer helices, forming a bend. Beta sheets are formed by H bonds between an average of 5 to 10 consecutive amino acids in one portion of the chain with another 5 to 10 farther down the chain. The interacting regions may be adjacent, with a short loop in between, or far apart, with other structures in between. Every chain may run in the same direction to form a parallel sheet, every other chain may run in the reverse chemical direction to form an anti-parallel sheet, or the chains may be parallel and anti-parallel to form a mixed sheet. The pattern of H bonding is different in the parallel and anti-parallel configurations. Each amino acid in the interior strands of the sheet forms 2 H bonds with neighboring amino acids, whereas each amino acid on the outside strands forms only 1 bond with an interior strand. Looking across the sheet at right angles to the strands, more distant strands are rotated slightly counterclockwise to form a left-handed twist. The C-alpha atoms alternate above and below the sheet in a pleated structure, and the R-side groups of the amino acids alternate above and below the pleats. The phi and psi angles of the amino acids and sheets vary considerably in one region of the Ramachandran plot. It is more difficult to predict the location of beta sheets than of alpha helices. The situation improves somewhat when the amino acid variation in multiple sequence alignments is taken into account. Loop. Loops are regions of a protein chain that are 1, between alpha helices and beta sheets, 2, of various lengths and 3-dimensional configurations, and 3, on the surface of the structure. Hairpin loops that represent a complete turn in the polypeptide chain joining 2 antiparallel beta strands may be as short as 2 amino acids in length. Loops interact with the surrounding aqueous environment and other proteins. Because amino acids in loops are not constrained by space and environment as are amino acids in the core region, and do not have an effect on the arrangement of secondary structures in the core, more substitutions, insertions, and deletions may occur. Thus, in a sequence alignment, the presence of these features may be an indication of a loop. The positions of introns in genomic DNA sometimes correspond to the locations of loops in the encoded protein. Loops also tend to have charged and polar amino acids and are frequently a component of active sites. A detailed examination of loop structures has shown that they fall into distinct families. Coils. A region of secondary structure that is not an alpha helix, a beta sheet, or a recognizable turn is commonly referred to as a coil. Protein classification. Proteins may be classified according to both structural and sequence similarity. For structural classification, the sizes and spatial arrangements of secondary structures described in the above paragraph are compared in known 3-dimensional structures. Classification based on sequence similarity was historically the first to be used. Initially, similarity based on alignments of whole sequences was performed. Later, proteins were classified on the basis of the occurrence of conserved amino acid patterns. Databases that classify proteins by one or more of these schemes are available. In considering protein classification schemes, it is important to keep several observations in mind. First, 2 entirely different protein sequences from different evolutionary origins may fold into a similar structure. Conversely, the sequence of an ancient gene for a given structure may have diverged considerably in different species while at the same time maintaining the same basic structural features. Recognizing any remaining sequence similarity in such cases may be a very difficult task. Second, 2 proteins that share a significant degree of sequence similarity either with each other or with a third sequence also share an evolutionary origin and should share some structural features also. However, gene duplication and genetic rearrangements during evolution may give rise to new gene copies, which can then evolve into proteins with new function and structure. Terms used for classifying protein structures and sequences. The more commonly used terms for evolutionary and structural relationships among proteins are listed below. Many additional terms are used for various kinds of structural features found in proteins. Descriptions of such terms may be found at the CATH website, the Structural Classification of Proteins, SCOP, website, and a Glaxo Welcome tutorial on the Swiss Bioinformatics XPASI website. Secondary structure. Secondary structure prediction is a set of techniques in bioinformatics that aim to predict the local secondary structures of proteins based only on knowledge of their amino acid sequence. For proteins, a prediction consists of assigning regions of the amino acid sequence as likely alpha helices, beta strands, often noted as extended conformations, or turns. The success of a prediction is determined by comparing it to the results of the DSSP algorithm, or similar, for example stride, applied to the crystal structure of the protein. Specialized algorithms have been developed for the detection of specific well-defined patterns such as transmembrane helices and coiled coils in proteins. The best modern methods of secondary structure prediction in proteins reach about 80% accuracy. This high accuracy allows the use of the predictions as feature-improving fold recognition and ab initio protein structure prediction, classification of structural motifs, and refinement of sequence alignments. The accuracy of current proteins' secondary structure prediction methods is assessed in weekly benchmarks such as LibBench and EVA. Background: early methods of secondary structure prediction, introduced in the 1960s and early 1970s, focused on identifying likely alpha helices and were based mainly on helix coil transition models. Significantly more accurate predictions that included beta sheets were introduced in the 1970s and relied on statistical assessments based on probability parameters derived from known solved structures. These methods, applied to a single sequence, are typically at most about 60 to 65% accurate, and often underpredict beta sheets. The evolutionary conservation of secondary structures can be exploited by simultaneously assessing many homologous sequences in a multiple sequence alignment, by calculating the net secondary structure propensity of an aligned column of amino acids. In concert with larger databases of known protein structures and modern machine learning methods such as neural nets and support vector machines, these methods can achieve up to 80% overall accuracy in globular proteins. The theoretical upper limit of accuracy is around 90%, partly due to idiosyncrasies in DSSP assignment near the ends of secondary structures, where local conformations vary under native conditions but may be forced to assume a single conformation in crystals due to packing constraints. Limitations are also imposed by secondary structure prediction's inability to account for tertiary structure. For example, a sequence predicted as a likely helix may still be able to adopt a beta strand conformation if it is located within a beta sheet region of the protein and its side chains pack well with their neighbors. Dramatic conformational changes related to the protein's function or environment can also alter local secondary structure. Historical perspective. To date, over 20 different secondary structure prediction methods have been developed. One of the first algorithms was Cho-Fossman method, which relies predominantly on probability parameters determined from relative frequencies of each amino acid's appearance in each type of secondary structure. The original Cho-Fossman parameters, determined from the small sample of structures solved in the mid-1970s, produce poor results compared to modern methods, though the parameterization has been updated since it was first published. The Cho-Fossman method is roughly 50 to 60% accurate in predicting secondary structures. The next notable program was the Gore method, named for the three scientists who developed it. Garnia, Osguthorpe, and Robson, is an information theory-based method. It uses the more powerful probabilistic technique of Bayesian inference. The Gore method takes into account not only the probability of each amino acid having a particular secondary structure, but also the conditional probability of the amino acid assuming each structure given the contributions of its neighbors. It does not assume that the neighbors have that same structure. The approach is both more sensitive and more accurate than that of Cho and Fossman because amino acid structural propensities are only strong for a small number of amino acids such as proline and glycine. Weak contributions from each of many neighbors can add up to strong effects overall. The original Gore method was roughly 65% accurate and is dramatically more successful in predicting alpha helices than beta sheets, which is frequently mispredicted as loops or disorganized regions. Another big step forward was using machine learning methods. First, artificial neural networks methods were used. As a training sets, they use solved structures to identify common sequence motifs associated with particular arrangements of secondary structures. These methods are over 70% accurate in their predictions, although beta strands are still often underpredicted due to the lack of three-dimensional structural information that would allow assessment of hydrogen bonding patterns that can promote formation of the extended conformation required for the presence of a complete beta sheet. CIPRED and JPRED are some of the most known programs based on neural networks for protein secondary structure prediction. Next, support vector machines have proven particularly useful for predicting the locations of turns, which are difficult to identify with statistical methods. Extensions of machine learning techniques attempt to predict more fine-grained local properties of proteins, such as backbone dihedral angles in unassigned regions. Both SVMs and neural networks have been applied to this problem. More recently, real-value torsion angles can be accurately predicted by SpineX and successfully employed for ab initio structure prediction. Other improvements. It is reported that in addition to the protein sequence, secondary structure formation depends on other factors. For example, it is reported that secondary structure tendencies depend also on local environment, solvent accessibility of residues, protein structural class, and even the organism from which the proteins are obtained. Based on such observations, some studies have shown that secondary structure prediction can be improved by addition of information about protein structural class, residue accessible surface area and also contact number information. Tertiary structure. The practical role of protein structure prediction is now more important than ever. Massive amounts of protein sequence data are produced by modern large-scale DNA sequencing efforts such as the Human Genome Project. Despite community-wide efforts in structural genomics, the output of experimentally determined protein structures, typically by time-consuming and relatively expensive X-ray crystallography or NMR spectroscopy, is lagging far behind the output of protein sequences. The protein structure prediction remains an extremely difficult and unresolved undertaking. The two main problems are calculation of protein free energy and finding the global minimum of this energy. A protein structure prediction method must explore the space of possible protein structures, which is astronomically large. These problems can be partially bypassed in comparative or homology modeling and fold recognition methods, in which the search space is pruned by the assumption that the protein in question adopts a structure that is close to the experimentally determined structure of another homologous protein. On the other hand, the de novo or ab initio protein structure prediction methods must explicitly resolve these problems. The progress and challenges in protein structure prediction has been reviewed in Jung 2008. Before modeling. Most tertiary structure modeling methods, such as Rosetta, are optimized for modeling the tertiary structure of single protein domains. A step called domain parsing, or domain boundary prediction, is usually done first to split a protein into potential structural domains. As with the rest of tertiary structure prediction, this can be done comparatively from known structures or ab initio with the sequence only, usually by machine learning, assisted by covariation. The structures for individual domains are docked together in a process called domain assembly to form the final tertiary structure. Ab initio protein modeling. Energy and fragment-based methods. Ab initio or de novo protein modeling methods seek to build three-dimensional protein models from scratch, based on physical principles rather than, directly, on previously solved structures. There are many possible procedures that either attempt to mimic protein folding or apply some stochastic method to search possible solutions, i.e., global optimization of a suitable energy function. These procedures tend to require vast computational resources, and have thus only been carried out for tiny proteins. To predict protein structure de novo for larger proteins will require better algorithms and larger computational resources like those afforded by either powerful supercomputers, such as Blue Genome Grape 3, or distributed computing, such as Folding at Home, the Human Proteome Folding Project and Rosetta at Home. Although these computational barriers are vast, the potential benefits of structural genomics, by predicted or experimental methods, make ab initio structure prediction an active research field. As of 2009, a 50 residue protein could be simulated atom by atom on a supercomputer for 1 millisecond. As of 2012, comparable stable state sampling could be done on a standard desktop with a new graphics card and more sophisticated algorithms. A much larger simulation timescales can be achieved using coarse-grained modeling. Evolutionary covariation to predict 3D contacts. As sequencing became more commonplace in the 1990s, several groups used protein sequence alignments to predict correlated mutations, and it was hoped that these co-evolved residues could be used to predict tertiary structure, using the analogy to distance constraints from experimental procedures such as NMR. The assumption is when single residue mutations are slightly deleterious, compensatory mutations may occur to restabilize residue-residue interactions. This early work used what are known as local methods to calculate correlated mutations from protein sequences, but suffered from indirect false correlations which result from treating each pair of residues as independent of all other pairs. In 2011, a different, and this time global statistical approach, demonstrated that predicted co-evolved residues were sufficient to predict the 3D fold of a protein, providing there are enough sequences available. Greater than 1,000 homologous sequences are needed. The method, if fold, uses no homology modeling, threading or 3D structure fragments and can be run on a standard personal computer even for proteins with hundreds of residues. The accuracy of the contacts predicted using this and related approaches has now been demonstrated on many known structures and contact maps, including the prediction of experimentally unsolved transmembrane proteins. Comparative protein modeling. Comparative protein modeling uses previously solved structures as starting points, or templates. This is effective because it appears that although the number of actual proteins is vast, there is a limited set of tertiary structural motifs to which most proteins belong. It has been suggested that there are only around 2,000 distinct protein folds in nature, though there are many millions of different proteins. These methods may also be split into two groups. Side chain geometry prediction. Accurate packing of the amino acid side chains represents a separate problem in protein structure prediction. Top methods that specifically address the problem of predicting side chain geometry include ident elimination and the self-consistent mean field methods. The side chain conformations with low energy are usually determined on the rigid polypeptide backbone and using a set of discrete side chain conformations known as rotamers. The methods attempt to identify the set of rotamers that minimize the model's overall energy. These methods use rotamer libraries, which are collections of favorable conformations for each residue type in proteins. Rotamer libraries may contain information about the conformation, its frequency, and the standard deviations about mean dihedral angles, which can be used in sampling. Rotamer libraries are derived from structural bioinformatics or other statistical analysis of side chain conformations in known experimental structures of proteins, such as by clustering the observed conformations for tetrahedral carbons near the staggered 60 degrees, 180 degrees, minus 60 degrees values. Rotamer libraries can be backbone independent, secondary structure dependent, or backbone dependent. Backbone independent rotamer libraries make no reference to backbone conformation, and are calculated from all available side chains of a certain type. For instance, the first example of a rotamer library, done by Ponder and Richards at Yale in 1987. Secondary structure-dependent libraries present different dihedral angles and/or rotamer frequencies for alpha, \alpha, helix, beta, \beta, sheet, or coil secondary structures. Backbone-dependent rotamer libraries present conformations and/or frequencies dependent on the local backbone conformation as defined by the backbone dihedral angles phi, \phi, and psi, \psi, regardless of secondary structure. The modern versions of these libraries as used in most software are presented as multidimensional distributions of probability or frequency, where the peaks correspond to the dihedral angle conformations considered as individual rotamers in the lists. Some versions are based on very carefully curated data and are used primarily for structure validation, while others emphasize relative frequencies in much larger data sets and are the form used primarily for structure prediction, such as the Dunn-Bragg rotamer libraries. Side chain packing methods are most useful for analyzing the protein's hydrophobic core. Where side chains are more closely packed, they have more difficulty addressing the looser constraints and higher flexibility of surface residues, which often occupy multiple rotamer conformations rather than just one. Prediction of structural classes. Statistical methods have been developed for predicting structural classes of proteins based on their amino acid composition, pseudo-amino acid composition and functional domain composition. Secondary structure prediction also implicitly generates such a prediction for singular domains. Quaternary structure. In the case of complexes of two or more proteins, where the structures of the proteins are known or can be predicted with high accuracy, protein-protein docking methods can be used to predict the structure of the complex. Information of the effect of mutations at specific sites on the affinity of the complex helps to understand the complex structure and to guide docking methods. Software. A great number of software tools for protein structure prediction exist. Approaches include homology modeling, protein threading, ab initio methods, secondary structure prediction, and transmembrane helix and signal peptide prediction. Some recent successful methods based on the CASP experiments include I-TASSER and Hilpred. For complete list, see main article. Evaluation of automatic structure prediction servers. CASP, which stands for Critical Assessment of Techniques for Protein Structure Prediction, is a community-wide experiment for protein structure prediction taking place every 2 years since 1994. CASP provides with an opportunity to assess the quality of available human, non-automated methodology, human category, and automatic servers for protein structure prediction, server category, introduced in the CASP 7. The CAMEO 3D continuous automated model evaluation server evaluates automated protein structure prediction servers on a weekly basis using blind predictions for newly released protein structures. CAMEO publishes the results on its website. CAMEO publishes the results on its website. CAMEO publishes the results on its website. CAMEO publishes